Hi, my name is Cameron Burke, and welcome to this Kaplan MCAT Science Review for DNA structure. This video covers the basics of DNA that you'll need to understand in order to be able to follow along about nucleotides. And the way that nucleotides can be classified is purines and pyrimidines. Additionally, you'll be introduced to the role bonding plays in the sugar-phosphate backbone, as well as the role it plays in denaturation and annealing. The learning goal for this video is to be able to recognize the molecules that join to create DNA and to describe their properties. By the end, you should be able to answer questions like this one about the sequences of nucleotides that create the most stability in DNA. DNA is a definitive characteristic of living organisms. It's the molecule responsible for providing genetic instructions that allows for the creation of all gene products. DNA is a two-stranded helical structure with an outer sugar-phosphate backbone and nitrogenous base pairs inside the helix. There are only 4 base pairs present in DNA: adenine, thymine, cytosine, and guanine. Adenine and thymine will always be paired, and cytosine is always paired with guanine. Knowing this will be helpful on test day because it means that amounts of adenine and thymine are equal, as are amounts of cytosine and guanine. This concept, also called Shulgarakh's rule, is important to keep in mind on the MCAT. There are only 3 different types of molecules that form DNA. The first category is nitrogenous bases, introduced a moment ago. For DNA, you only need to worry about adenine, thymine, guanine, and cytosine. The second type of molecule is a pentose sugar, which connects the nitrogenous base to the third type of molecule, a phosphate group. When wound into a double helix, the nitrogenous bases form a core inside the sugar-phosphate backbone. Now that we've seen how the 3 molecules form DNA as a whole, we can move on to the polymers they create interacting with each other individually. Nucleosides are the smallest polymer within DNA. Nucleosides consist of a nitrogenous base bound to a pentose sugar only, without the phosphate group. When the phosphate is also considered, the polymer is called a nucleotide. Together, nucleotides bind to create strands of DNA. The pentose sugar in each nucleotide determines whether the nucleic acid in question is DNA or RNA. Ribonucleic acid, or RNA, has a ribose sugar. Ribose can be identified by the presence of 2 alcohol groups. DNA, or deoxyribonucleic acid, has only 1 alcohol group present. The absence of the second alcohol is one clue to look for on test day to help you identify if you're looking at DNA or RNA. DNA is the one with only 1 alcohol. Ribose or deoxyribose is one way to identify nucleotides, but another way to identify them is by determining if they are purines or pyrimidines. Adenine and guanine are the only 2 purines, as determined by the presence of 2 rings in their structure. On the MCAT, the mnemonic pure as gold can help you remember which bases are purines. Pyrimidines only have 1 ring. The mnemonic to help you remember that is that pi is only 1 circle. Cytosine, uracil, and thymine are all pyrimidines. The mnemonic to remember this is cut the pi. However, on test day, it's up to you to remember that RNA has uracil instead of thymine. Because both purines and pyrimidines have nitrogen in their rings, they can be described as heterocycles. Biological aromatic heterocycles obey the typical rules for aromaticity. Because they're cyclic, planar, conjugated, and they obey Huckel's rule, they count as being aromatic. However, because purines and pyrimidines contain nitrogen, the nitrogenous bases are classified as heterocycles. It's pretty vital that nucleotides are unreactive. Aromaticity creates very stable conditions through electron delocalization. For this reason, biological aromatic heterocycles are an ideal foundation for DNA. Now that you've been exposed to nucleotides, we can move on to the way they're organized into a chain by a sugar-phosphate backbone. In the same way that you read words left to right, you read a single strand of DNA from the 5' end to the 3' end, with the nitrogenous bases aligned in the middle. The polarity of DNA is determined by the negatively charged phosphate group. The 5' end has a negative charge, while the 3' end has an uncharged alcohol group. DNA is anti-parallel, meaning that one strand is read in the 5' to 3 direction, while the other complementary strand is in the 3' to 5' direction. Double-stranded DNA forms due to the energetic favorability of hydrogen bonds forming between base pairs. Cytosine and guanine share 3 hydrogen bonds, while adenine and thymine share 2 hydrogen bonds. Because it takes more energy to break higher numbers of bonds, DNA with a larger percentage of cytosine-guanine bonds will be more stable. The process of breaking hydrogen bonds that connect 2 strands of DNA is called denaturing. Because DNA is just a series of molecules shaped into a double strand through hydrogen bonds, it can be denatured by anything that disturbs the hydrogen bonds. On test day, heat is the main cause of denaturation to look for, but extreme pH changes or chemical influence can also separate double-stranded DNA. It's important to note that denaturation is specifically referring to breaking hydrogen bonds between base pairs, not breaking the bonds that involve the ribose sugar or the phosphate. The process of reversing denaturation, or creating hydrogen bonds between two strands of DNA, is called annealing or hybridization. Annealing can be induced by cooling strands of DNA, removing energy to make hydrogen bonding more favorable. Many pieces of biotechnology that you may be asked about on test day rely on denaturing and annealing DNA to produce effective results. Now you have the opportunity to see how you're doing with the learning goal for this video. The first practice question tests your knowledge of DNA stability. The correct answer is choice C, because the strand with the highest percentage of cytosine-guanine pairs will have the most hydrogen bonds and thus have the greatest stability. The second practice question addresses your knowledge of ATP. The correct answer is C. Adenosine triphosphate, or ATP, is a high-energy molecule because the negatively charged phosphate groups want to get away from each other. To recap, a nucleoside and a phosphate form a nucleotide. Nucleotides form a sugar-phosphate backbone, creating a single strand of DNA. Two single strands of DNA form a double helix. Adding heat can separate the double helix, causing it to denature into a single strand of DNA. Thanks so much for watching this Kaplan MCAT science review about DNA structure, and good luck when you take the MCAT!